A very good morning to all of you. So in today's video, I will just share the must read topics. Although in MD exam it's not possible to say the which topic is very important. First thing, you have to cover all the each and everything which is written in your NMC syllabus. Okay, in paper 1, paper 2, paper 3 and your paper 4. And I hope all of you having an idea what is your NMC curriculum syllabus. And those who are now first year JRs resident, if you didn't get your syllabus, then it's easy for you. You can go through the NMC site and in the Google, and in Google you will just type the NMC syllabus of biochemistry. From that you can easily get the syllabus of your MD curriculum. Alright. So in today, because of the request of a lot of my juniors, ma'am please share some must read topics. So that's why I'm just making this video. Otherwise it's not easy to say. You have to read each and everything in your syllabus. But at the last end or last moment of the exam, it's not possible to read or revise everything. It's a genuine. This thing happened with all of us. Okay. So at least before 15 days of your exam. So there are some must read topics. That is, these topics you cannot skip in your exam while you are going to do or preparing for your university exam. So this topic you must see before your exam. Alright. So today we will discuss paper 1 and paper 2, and in the next video we will discuss paper 3 and paper 4. Alright. So let's start it. So in paper 1, first is biomolecules. And biomolecules, first is that of your proteins. So important thing in proteins is that of at MD, that is of what is the long question it is asked is what is the structural organization of proteins. Alright. The first thing you have to study is that of structural organization of protein. Then short note is biologically important peptides. Okay. Next question is asked, that is structure function relationship of proteins like your hemoglobin, myoglobin, collagen and immunoglobulin. So you should have a basic idea what is the structure function relationship of these proteins. Next is your important topic, protein folding and its associated disorder. Okay. Next short note is that of protein denaturation. Sometimes protein denaturation asked as a short note. Okay. Then protein degradation, that is lysosomal degradation and proteasomal degradation. And question can be asked as a short note, that is lysosomal degradation, or can be asked as a short note what is proteasomal degradation. And sometimes one additional question is added in it, what is the difference between lysosomal degradation and what is difference in between lysosomal and proteasomal degradation. So you have to prepare according to that. Okay. Then comes on the plasma proteins. Plasma protein an important topic, and you must have an idea regarding the electrophoretic pattern of different kind of in different kind of diseases. Like what is the electrophoretic pattern of plasma proteins? You will get in nephrotic syndrome, in cirrhosis of liver, in your multiple myeloma. So you should know, and it is also important for your viva also. So plasma protein is one of the important topic. Okay, and especially your electrophoretic pattern. Alright, and albumin, what is the function of albumin that you have to know. Alright, now comes on the carbohydrate. In carbohydrate, that is chemistry. First paper includes your chemistry. Okay, so in carbohydrate chemistry, the important thing is that of mucopolysaccharide. You know each and everything for mucopolysaccharides or GAPs, including mucopolysaccharidosis. What are different types of mucopolysaccharidosis? Okay, it's an important topic. Then other thing you must read, that is the glycation and glycosylation. What is difference between glycation and glycosylation? You have to know. Okay, and what is the role of carbohydrate in blood group substance? Okay, so this is an important topic in carbohydrate chemistry. And one important short note is that of asked as a reasoning like question, that is why sucrose is non-reducing sugar? Explain. Next question is why sucrose is known as invert sugar? Explain. So these can be asked as a reasoning type of question. Okay. And for short note in chemistry is important is your mutarotation. What is the mutarotation and what is the clinical significance of mutarotation? Okay. So this thing you have to must read. Okay. Now comes on the lipid chemistry. Lipid chemistry, you should know the lipid classification. What is the definition of a lipid and what is the classification of lipid? Okay. And then you have to know fatty acids. What is the nomenclature, classification and your properties of fatty acids. And important thing is that of short note on essential fatty acids. What is essential fatty acids? Okay, you have to know, and essential fatty acid acts as a short note also. Okay, and short note, another short note is that of trans fat. Trans fat is also important as your short note. Now comes on the cholesterol. Cholesterol important as that what is the structure of cholesterol, what is the properties and biomedical importance of your cholesterol. Okay. Next is that of phospholipids. Phospholipid is important for your 10 marks question. That is, what is phospholipid, what is defined classification and properties of phospholipid, and what is the biomedical importance of various phospholipids. So you have to prepare according to that. Okay. Next comes your lipoproteins. What lipoproteins, important thing is that of your classification and properties of different type of your lipoproteins, and they are importance in health and disease. Okay. Next is that of structure, properties and biomedical applications of your micelles and liposomes. Important as a short note. And also you have to prepare what is the difference between micelles and liposome, because in one of the university exam, in previous university exam, the question can be asked as a difference between micelles and liposome. Okay, and what is the biomedical importance of your micelles and liposome. Okay, so this is about your lipid chemistry. Now comes on nucleotides and nucleic acid. So you should have a basic idea what is difference between nucleosides and nucleotides. Basic idea in Viva also. Okay, so this not can be asked in a theory, but for Viva you have to know what is difference between nucleosides and nucleotides. Okay, so short note in this topic is that of biologically important nucleotides. It is asked as a short note. Another important is that of synthetic analog of purine and pyrimidines. Asked as a short note. Okay. And next in this topic is that of different types of RNA. Short note can be asked what are the different types of RNA, and short note on Watson and Crick model of DNA. Okay, so this is all about your chemistry of nucleosides and nucleic acid. Okay, now comes on the cell biology. So cell biology, the important thing is that of structural organization and function of biological cell and different subcellular organelle along with their marker enzymes. It can be asked as a LAQ also. So you should know what is the structural organization. Okay, that is you have to know about, you have a basic idea about what is fluid mosaic model, and simply fluid mosaic model is asked as a short note also individually. Fluid mosaic model or LAQ can be asked as a structural organization and biological functions of biological cell, and what are the different subcellular marker with their with subcellular organelle along with their marker enzyme. And sometimes in one of the previous exam also, a short note simply asks what is the different biomarker enzymes of subcellular organelle, and it is also important for your Viva. So you should know about that. Next thing important in this topic of cell biology is that of cell fractionation and separation of organelle. Okay, what is cell fractionation and how the separation of organelle will done. Okay. Next thing is that of shorting of protein. Protein shorting. Protein shorting is also known as protein targeting. Okay, it's an important topic. Protein targeting or protein shorting. Okay. And what are the disorders associated with the protein targeting. Next is that of important thing you have to study is disorders related to cell membrane and subcellular organelle. Okay, that is disorders related to your cell membrane and subcellular organelle. Alright. Next comes on the extracellular matrix. Extracellular matrix, you have a basic idea how collagen is synthesized in your body and what are the disorders which are associated with the collagen. Okay, so this thing you have to know in extracellular matrix. Okay, next important thing is that of components of cytoskeleton. Okay, the question can be asked what are the components of cytoskeleton and what is their role in muscle contraction and cell motility. So you must know. Okay. And then comes on the cell cycle. The question asks as of what is cell cycle and how the cell cycle is regulated. That is cell cycle regulator. LAQ can be asked as of what is cell cycle, define or describe different phases of cell cycle. Okay, like prophase, metaphase, and you have to describe that, and how the cell cycle is regulated. Or sometimes simply a short note can be asked individually, cell cycle regulators. How the cell cycles are regulated. So you must read this topic. And next important topic is that of mechanism of cell death, that is your apoptosis. Okay, short note apoptosis is important for your short note. Okay, I already shared a video on apoptosis, so you can go through that. Okay. And next come on the paper one is that of fluid electrolyte and acid-base balance. In acid-base balance, each and everything is important. So you cannot skip anything. You must read what is metabolic acidosis, alkalosis, all the causes, okay, and all the compensation. What is the compensatory mechanism? And respiratory acidosis, respiratory alkalosis, what are the causes and what is the compensatory mechanism? And important short note in that topic is that of anion gap and what is the significance of anion gap. Okay, so in acid-base balance, this thing you have to know. And in some universities also, like case-based questions also asked. So in this case-based question, you have to know question will come like PCO2, pH and bicarbonate value will be there, and question will be asked to find out whether it's what kind of acidosis or alkalosis will there. Okay, so you also prepare accordingly. Okay. Now comes our analytical techniques and your instrumentation. So in analytical techniques and instrumentation, important is your colorimetry, spectrophotometry, atomic absorption spectrophotometry, your flame photometry, fluorometry, turbidimetry and nephelometry. Okay. And then in electrochemistry, important thing is that of ion selective electrode. Okay. Ion selective electrode is important. Okay. And other things you can skip, but ion selective electrode is must read topic. Okay. And important is your biosensors. Biosensor is also important, biosensors. Okay. And then electrophoresis. Electrophoresis asks as a long question. What is electrophoresis? What are different types of electrophoresis? Is application. And in this you must know individually, like SGS page, isoelectric focusing, capillary electrophoresis. Okay. And 2D electrophoresis. Okay. So these types of electrophoresis you read thoroughly. Okay. Now comes on the chromatography. It can be asked as a LAQ. What is chromatography? Okay. How this technique is performed? What are the application of chromatography? Or sometimes individually asked as a HPLC. HPLC individually asked as a short note. Okay. And gas chromatography. Additionally, besides HPLC, other things you have to read is that of gas chromatography and affinity chromatography. So HPLC, gas chromatography and affinity chromatography. These are the must read topics of in your chromatography. Okay. Next come on the nanotechnology. Nanotechnology is important for your short note. It can be asked frequently in various exams as a nanotechnology in the short note. So you should know what is nanotechnology and what is the applications of your nanotechnology. Okay. Then in techniques to study in vivo metabolism, important is that of NMR. Okay, NMR is very important topic. NMR and in some exam PET scan is also asked. So NMR and PET scan, PET scan, these two topics you must read. Next is that of automation. Automation is very important. What is automatic analyzer and what is semi-automatic analyzer? What is fully automated analyzer? So automation is also one of the important topic. Okay, and next is that of POCT, that is point of care testing. Okay, point of care testing, this is an important topic. And in immunochemical techniques, you must read your ELISA. What is ELISA? What are different types of ELISA and what is the application of ELISA? And in PCR, you know what is the conventional PCR, and important additional thing is that what is real-time PCR and what is the difference between conventional PCR and real-time PCR. And in previous exam, question is asked as a real-time is why real-time is known as real-time. Okay, and that should also know for the your vibe also. Okay, all right. Now comes on paper, another part is that of your biostat and research methodology. One question comes as a short note in this topic. Okay, biostat and research methodology. So important thing is that of you must study different type of statistical tests like t-test, ANOVA, that is analysis of variance, chi-square test, and what is difference between parametric test and non-parametric test, and what is correlation and regression. And in one exam, the question is asked, what is the difference between correlation and regression? Okay. On my channel, I have already continued the biostat series and few videos I have already uploaded. So you can go through that. Okay. So these are the statistical tests. Another is type of study design. What are different type of study design and sampling methodology. Okay. Next is that of important thing is that of statistical methods of validation of diagnostic test. Okay. So frequently asked question is various exam, that is statistical methods of validation of diagnostic test. Okay. Another is that of meta-analysis and systematic review. It's very important, meta-analysis and systematic review. A lot of times question is asked. What is meta-analysis and systematic review? Write a note. Okay. An important thing is that of ethical aspects related to research and publication. It is also asked in one or two exams. That is, what is the ethical aspects that is related to your research and publication. Okay. And what is electronic search of literature. Okay, so these are the must-read topics in biostat part of your paper 1. Okay, now in coming scenario of one question can also be asked from your medical education. Okay, so important topics that you have to cover in medical education is that of microteaching, short note, microteaching and your reflection writing and OSPE. Okay, microteaching, reflection writing, OSPE. In OSPE, the question is asked what is OSPE and what is difference between OSCE and OSPE. OSCE you will do in your clinical scenario and OSPE you will do in non-clinical. So you must read what is OSPE individually. OSPE is asked as a short note, and what is difference between OSPE and OSCE. And what is the principle of self-directed learning, that is SDL and giving feedback. Okay, answer the short note, that is what is the principle of SDL and its feedback. And one important short note is that of ATCOM. ATCOM, AETCOM, ATCOM. ATCOM acts as a short note. So this is all about your paper 1, and these topics are that you cannot skip before your exam. So these are the must read topics in your paper 1 before your university exam. Alright, so in next video we will discuss a paper 2. Okay, so thank you.